Okay, let's unpack this. We're diving deep into an idea that sounds, well, almost counterintuitive.
Definitely. Using viruses to fight cancer.
Yeah, the very things we usually try to avoid. It's called oncolytic virotherapy, or
Yeah.
OVT.
That's it. And we've got this great stack of research tracing its whole history.
From really ancient observations all the way up to, you know, modern approved drugs.
Exactly. So our mission here is to follow that journey, pull out the key moments, the surprising facts.
The breakthroughs that turned viruses from, well, foe
Yeah.
to potential friend in cancer treatment.
Right. And it's such a big deal now. Some are calling OVT a fifth pillar of cancer therapy.
Alongside surgery, chemo, radiation, targeted therapy. Wow.
It's getting there.
What gets me is how far back the basic idea seems to go. You mentioned sources hinting at tumor shrinkage after fevers or infections way back in ancient Egypt.
Yeah, really early anecdotal stuff. And things like variolation, so, using a bit of smallpox to prevent the severe disease.
Uh-huh. Shows people were thinking about manipulating host pathogen interactions for health, way before we even knew what viruses were.
It started just by watching, observing. Then you fast forward to the late 19th, early 20th century.
When medical science was really starting to take off.
Exactly. You had pioneers like William Coley noticing this link between infections and cancer regression.
Ah, yes, Coley's toxins. Using bacterial products to try and stimulate an immune response against tumors, right? An early kind of immunotherapy.
Pretty much. And around the same time, we see the first documented cases where viruses seem to shrink tumors directly.
But the first deliberate attempts, maybe mid-20th century, using viruses, they didn't really pan out initially.
No, they hit some major walls. The technology just wasn't there yet. It was hard to control the viruses reliably.
So safety concerns, consistency issues.
Yeah, both. So OVT sort of stalled for a bit, but then the later 20th century happened.
And boom, the game changers you highlighted. Immunology and molecular biology.
Absolutely. First, just this explosion in understanding fundamental immunology, how T cells work, discovering cytokines, immune checkpoints.
It's like CTLA-4 and PD-1, the brakes on the immune system.
Exactly. Understanding how cancer can use those brakes. And then, just as critical.
The recombinant DNA revolution, being able to cut and paste genes.
Yes. The molecular scissors and glue from Berg, Boyer, Cohen, and others, that changed everything for OVT.
How so? What did that enable?
It meant scientists could move from just, finding viruses that seemed to like cancer cells and hoping for the best.
To actually designing them, engineering them.
Precisely. You could rationally engineer them. Delete genes that cause disease in normal cells, enhance tumor activity, even add new genes.
Like adding instructions for the immune system.
You got it. It shifted OVT from empirical selection to rational design. Much safer, much more potential.
And that engineering work led directly to the first regulatory approvals, TVEC being the big one in the US and Europe for melanoma.
Right, TVEC or Imlegic. It's a herpes simplex virus, HSV1 as the backbone.
But heavily modified, yeah.
Oh, yes. Key genes deleted for safety, like ICP34.5, so it doesn't replicate well in healthy cells. Another deletion, ICP47, helps tumor antigens get presented better to the immune system.
And it's armed, isn't it? That's a key term.
It is. They added the gene for GM-CSF. Think of it like a beacon.
Calling in the immune cavalry.
Exactly. It recruits and activates immune cells right there at the tumor. The big trial, OP-TIM, showed it improved durable responses, especially in earlier melanoma stages.
And it showed a systemic effect, not just local killing.
Crucial point. It proved boosting the immune response was key.
Before TVEC, though, China actually had the very first approval, H1N1.
That's right, way back in 2005. H1N1 is an engineered adenovirus. It targets cells defective in the p53 pathway, which is common in many cancers.
Approved for head and neck cancer, used with chemo.
Yes, a major milestone.
And then more recently, G47 in Japan.
Right, DeliTact, another engineered HSV1, but kind of a third-generation design. Multiple deletions focusing on safety, targeting rapidly dividing cells, and enhancing that immune presentation.
And approved for malignant glioma based on impressive Phase 2 survival data.
Conditional approval, yes, for a really tough brain cancer. These approvals, they're huge validation. OVT is real.
So it's crystal clear now, it's not just the virus physically bursting the cancer cell. It's this dual action.
That's the modern understanding, absolutely. Direct oncolysis, the virus killing the cell, releases tumor antigens. Danger signal.
Wakes up the immune system.
Exactly. And the viral infection itself triggers innate immune responses. Together, it can turn these immunologically cold tumors
The ones the immune system ignores.
into hot tumors that get recognized and attacked. It's a powerful 1-2 punch.
Which explains why combining OVs with checkpoint inhibitors is such a hot area now. Take the brakes off the immune system and flag the tumor.
Precisely. There's a really strong rationale for synergy there. Unleash the immune system that the virus helped prime.
It really feels like we've come full circle from those ancient inklings about infection through massive scientific leaps.
To now, where we're engineering these viruses with incredible precision.
But it's not perfect yet. What are the hurdles still being tackled? You mentioned delivery.
Yeah, getting the virus effectively to all tumor sites, especially metastases, that's still a challenge. Systemic delivery.
And the body's own immune system fighting off the virus before it can work.
That too. Pre-existing or developing antiviral immunity can limit repeat dosing or effectiveness. Finding biomarkers to predict who responds best is another big area.
But the engineering toolbox keeps getting bigger.
It does. New ways to shield the virus, new types of viruses, more complex payloads they can carry. The field is moving fast.
Which kind of leads to a final thought then. Looking ahead, could you see these tailored oncolytic viruses becoming a really fundamental part of personalized cancer treatment?
I think that's the direction. Imagine engineering viruses not just with one immune booster, but maybe multiple signals tailored to a patient's specific tumor and immune profile.
Or combining different OVs or OVs with other therapies in really smart sequences.
Exactly. Using them as a versatile platform. It certainly feels like OVT has cemented its place and its potential is still unfolding.